Everyone, welcome to Global Academy Institute Technology Foundation Incorporated. Our topic for second quarter, week 4, is genetic engineering, genetically modified organisms. Genetic engineering is the process by which pieces of DNA is transferred from one organism to another. A vector is needed to transfer a gene into a host cell. Vector, plasmids or viruses are vectors. A plasmid is a circular piece of DNA in bacterium. What is DNA? Deoxyribonucleic acid is a large biomolecule that contains the complete genetic information for an organism. The basic building block of DNA molecule is called a nucleotide. A single strand of DNA may contain billions of nucleotides. Refer to Figure 1 to see the DNA structure with labeled parts. Although each DNA molecule contains many of these building blocks, only 4 unique nucleotides are used to create the entire DNA sequence. These are written as A, G, C, and T. Analogous to how the 26 letters of the alphabet can be arranged to create words with different meanings, these 4 nucleotides can be arranged in sequences to spell the genetic instructions to create all of the different proteins organisms need to live. Because DNA contains instructions for an organism to create several different proteins, it is useful to define another subunit of DNA called genes, shown in Figure 2. To use another analogy, think of the genome as an entire cookbook for an organism, and each gene is an individual recipe in that cookbook. When a single recipe is followed, the result is a specific protein. Why are proteins important? Proteins perform all of the work in organisms. Some functions of proteins include serving as catalysts for reactions, performing cell signaling, transporting molecules across membranes, and creating structure. Recombinant DNA technology. Changes in the DNA of an organism can cause a change in traits, and its manipulation could lead to the formation of genetically modified organisms. Recombination of DNA technology is the technique of combining 2 DNA sequences from different sources. One goal of this technique is cloning a particular gene for analysis or mass production of a medically useful protein product. Bacteria, yeast, and cultured plant and animal cells are usually used for recombinant DNA experiments. A quick look at the process. Number 1. DNA carrying a gene of interest is taken from a cell. Number 2. The gene is inserted into the DNA of another cell. 3. The host cell now contains recombinant DNA. 4. The host cell multiplies. And 5. The desired protein is produced. To make insulin for use by diabetic person before genetic engineering, where did insulin come from? Pigs and cows. Insulin produced by genetically modified bacteria. The human gene able to make insulin is inserted into bacteria. Number 2. To make growth hormone to treat dwarfs. 3. To prepare vaccine. 4. To make plant resistant to disease. And number 5. Genetically modified rice, rich in vitamin A. An organism which has foreign DNA integrated into its genetic material is called genetically modified organism. Possible hazards of genetic engineering. Risk for human health, high chances for cancer. Risk for animal health, higher chances of infection. And risk of creating new microbes that are difficult to kill by antibiotics. Thank you for listening, and God bless.